Hello and good afternoon. It's great to see such a full house. First, I want to thank Congressman Fattah for being with us today, and I would like to thank the Society for Neuroscience and the Optical Society for co-sponsoring this special event with the National Science Foundation. Representative Fattah's efforts have been instrumental in bringing brain science to the level of attention it's getting today. Congressman Fattah's innovative policy initiatives to intensify federal research efforts across brain disease, disorder, injury, cognition, and development have helped promote research and discovery in brain science. We are so pleased to have him here with us, and without further ado, may I introduce Congressman Chaka Fattah.
First and foremost, this is so appropriate that the foundation would host a briefing like this. We have an enormous treat for you for lunch. You're going to hear from a group of the leading scientists in the world on the most important topic. At least for me, it's superior among all other equal scientific areas of investigation. So neuroscience is number one, and we have a panel that you're going to be, I think, really pleased to hear from. So I'm going to be brief. But they're well over, by the World Health Organization's estimation, 1 billion people in our world suffering from some form of brain disease or disorder. The National Institutes of Health says more than 50 million Americans suffering from a brain illness. Beyond the question of disease and disorders, we have the questions around cognition and teaching and learning. We have a host of areas that we can learn. Neuroscience is a place in which, a field in which ignorance still outweighs knowledge. And there's so much more for us to learn. And that's why in this year's appropriations, we've given the foundation, I think, a healthy start to continue that work on $146 million additional dollars, a major increase over last year, an investment in the creation of the first ever U.S. National Brain Observatory and building a partnership with our national labs. And you couldn't have a better leader for this effort than Jim, since he comes out of the lab movement and Sandia. So there's a lot for us to do together, and there's even more with the International Conference on Neuroscience. But for today, you have plenty enough on the plate for lunch, and I'm going to step aside, and we're going to learn from our guests.
So as the Assistant Director for Biological Sciences at the National Science Foundation, and as a neuroscientist, it is gratifying to hear Congressman Fattah talk about the brain and the importance of cognitive science and neuroscience research. As you have heard, Congressman Fattah is not only passionate, but exceptionally knowledgeable about issues related to the brain. Thank you, Representative Fattah, for your inspiring words and efforts to promote understanding of brain science. Now, I'd like to take the opportunity to speak to you briefly about the National Science Foundation's efforts to support neuroscience. In 2012, Congress, with Representative Fattah's enthusiastic support, encouraged the National Science Foundation to create a cross-foundational activity in cognitive science and neuroscience. The Congressman recognized that the NSF was uniquely positioned to advance research in these areas, and the National Science Foundation responded by developing a cognitive science and neuroscience roadmap outlining our priority areas and potential funding mechanisms. Then, in fiscal year 2013, as you know, President Obama announced the Multi-Agency Brain Initiative, with NSF as one of the 3 lead agencies, along with the National Institutes of Health and DARPA. As a result, the Biological Sciences Directorate, in cooperation with other NSF directorates, has initiated Understanding the Brain, a unique program for fundamental research in neural circuits and neurotechnology, which draws together NSF's ongoing activities in cognitive science and neuroscience and new brain initiative activities. Understanding the Brain aims to generate the tools needed to explore healthy brain function and to establish a comprehensive understanding of how thoughts, memories, and actions emerge from the dynamic activities of the brain. Scientists who conduct fundamental research need the support and services of professional organizations, such as SFN and the Optical Society, to maximize their success and engage fully in the work that is transforming the field of neuroscience as we know it.
How can a machine that's constantly turning over its parts, constantly changing its bits and pieces, encode memories that can last for a lifetime? that's a mystery. Now, the philosopher scientist William James, at the end of the 19th century, put this rather nicely, I think. He writes, "The stream of thought flows on, but most of its segments fall into the bottomless abyss of oblivion. Of some, no memory survives the instant of their passage. Others, again, leave vestiges which are indestructible, and by means of which can be recalled as long as life endures." Can we explain these differences? Can we explain the difference between an ephemeral memory and one that endures as long as life? Well, for the 80 years after he posed that question, the answer was a definitive no. There was no idea at all, really, how it happened. But that's changed very much in the modern era. And part of the reason that changed was the discovery of what is probably the single most remarkable physiological effect found thus far in the brain. And I wanted to show you a little bit of this. Now, what you see here is a picture of a section through the brain rat. It's living in a dish, and it's bubbling along just fine. And you can see in there are two wires, those orange things. And between them, there's a sort of a pole. It's actually a pipette that records signals. Now, of course, what's actually down in there are the piece to the right, this neuron here. And there's several of them down there. There's actually dozens of those down there, all right? So how does this system work? It's something like a phone call. When you get a phone call, somebody calls, and there's a message that comes down a wire to a speaker, to a device, and then your ear acts as a receiver. Now, when the caller calls, you get that signal. What's happening in your ear, of course, is there's a sound. There's a certain loudness that's generated and then transported to the brain. This thing works something like that. The wires are making the call. The brain synapses, or sort of ears, are receiving the call, and we're picking that up with the wire, with the pipette. You can see here over there to the left, that's what the level of sound, of loudness, if you will, the synapses record when they get a routine call. But now we've discovered the brain has a trick no telephone system has. You can send a very brief 2-second code down the wire. When that code arrives, and you come back sometime later and make another call, you can see over there the signal is larger. It's as though that signal turned up the loudness of your phone. But here's the magic part. That doesn't go away. You can come back weeks and months later and make a call, and it's still louder. Somehow, that 2-second code changed permanently the strength of the synapses. Studies from around the world have convinced neuroscientists that what you're seeing there is, in fact, the substrate of commonplace memory. But, of course, there's another question. How can the brain's connections do a trick like this? And the problem's daunting because there's hundreds of thousands of synapses, even at the tip of that one pipette. How can it do that? And we couldn't get an answer without the development of new technology. This technology lets us zoom in to the level of the individual contacts. Those little green blobs you're seeing are synapses. And there's quite a large number of them. And we're just going through a field of them, actually, where the learning took place. And every so often, you'll see one of them is orange. That's a synapse that's been modified by the magic code. So we can actually see those things today. Led to many discoveries, and I can only have time to tell you about one. They're bigger. After the code comes, it physically changes the synapse, the connection, and makes it bigger. And that's why it's stronger. That's why the signal's louder. Now, scientists are never satisfied at this level. We've got to drill down inside one of those contacts that's been modified. We can't do that yet, but we're getting close. This is a picture at the top there from a technique called storm microscopy. This thing's a revolution. We've drilled inside one connection, and now what you're seeing is it's no longer orange, the modified contact. That's because the modifier proteins are red, and the synapse is green. And you put them together, and you see that orange blob. But you are actually visualizing the individual proteins inside one contact. I really expect that within a year, maybe a little longer, I'm always the optimist, there will be a movie of you seeing the proteins moving around in a contact after the the signal arrives to say, let's have modification. You'll see these guys running around. We'll have a movie for you. Give us a couple years, though. Now, oftentimes people watch this kind of thing and they get scientists drilling down. They're going down a rabbit hole that really doesn't have a bottom. And I understand that. But actually, at each one of these stages, there's spin-offs that mean a lot to people. What you're seeing now is a picture, autopsy material of the human brain, where we're using those methods to reconstruct hundreds of thousands, millions of synapses from people who have had devastating psychiatric illnesses. And so now we can finally answer the question, are the synapses too big for someone who's had major depression, or are they too small? And if you look at to your right side of the image, you can see we can even go into those contacts and look at those modifier proteins and all other kinds of proteins. And we can ask, are the modifiers overactive? Are they underactive? Now, this work is going on. This is all new. It's just starting. But already, laboratories around the country are looking at these conditions. And these are the only ones I know about. We're going to have a picture of the synapses from drug addiction. We're going to have a picture of synapses from schizophrenia. And when I say picture, I mean hundreds, millions of these things. So we can do real detailed measurements, finally. Now, I need to return to the chemistry of memory. And this now is because it's not enough for us simply to know which proteins are involved in changing those connections. We have to know the sequence in which they act. These things, these proteins are like pool balls. Now, I know many of you wouldn't know about pool. I assume congressmen won't know about pool, but it's a game. And the proteins are like pool balls. You hit one, bounces into another, makes a cascade, right? These are the proteins. This is the cascade. And I only want to say two things about it. You see where it's going at the bottom, down there? It leads to a change in the cytoskeleton, to the skeleton of the synapse, to the structure of the synapse, to the actual structure of the thing. And it changes the structure, and because it's structure, and then re-anchors it, that's why memory endures. Because the change in the end was structural. And that's the answer to William James's question. Now, at the top, you see over here, there's another point I want to tell you about. This whole process is driven by the brain's primary growth factor. What we're finding at this level is that growth and learning are intertwined. Okay? What I'm saying is that your growth processes are being used to stamp in your memories. And your learning is activating the growth processes. And I'll leave it to you to develop the implications of that. Okay, and again here we're seeing spin-offs. This is a list of 8 conditions for which memory problems are prominent. Now, we have rodent models for these 8 conditions. Labs around the country have shown all these models have a defect in that roadmap I showed you in the preceding slide. Every one of them has a defect, a different defect, but a defect. And we think they're all leading to a final endpoint failure in that business of restructuring your synapses. That's what we think is happening. It's a final failure in all of these conditions. Now, of course, knowing where the little defects are, we're no longer shotgunning in our treatment at these cognitive problems. Now we're pointing it precisely. And labs again, I always have to say labs again because I have a tendency to take credit for a lot of stuff. I mean, Dr. Wolves knows about that. When we were,
deserve credit.
Yeah. Labs around the country are now have successfully published papers saying they have rescued in the rodent models these 5. And how did they do it? With drugs that boost the brain's growth factor, primary growth factor. Now, I'm particularly interested in that third one on the list, and you can guess why. I know you're not particularly interested, but I am. You can see why I'm really interested. And we're doing studies now, as Jim mentioned, where we're trying to slow the atrophy of the brain by boosting growth factors. Now, my time is up. Now, I want to say only one note about where this is all going. We're now putting the synaptic chemistry back in brain networks. We're now asking, where is the memory occurring? We don't know. And because we don't know where the memory is, we can't build a theory of recall. And without a theory of recall, we can't build a theory of how memories are integrated into thought. But that's coming, because we're learning where the memories are. But I want to tell you more about networks, because these people are following me know a lot about them. Thank you so much.
Thanks, Dr. Lynch. Dr. Smith from UNC Chapel Hill.
All right, so we're going to move from memory to thought. My lab has recently invented the first microscope for flexibly imaging neural activity in multiple areas of the brain at the same time. This innovation, currently funded by the NSF, is important because it lets us explore how brain areas work together, and I'm very excited to share it with you today. The key thing we achieved is a broader field of view. We can now see neural activity over a larger portion of the brain than ever before. To understand the importance of this, recall the parable of blind men and the elephant. Blind men were asked to approach an elephant and identify what it is. The blind man who approached the trunk announced that the elephant is a new kind of snake. The blind man who approached a foot said that the elephant is a new kind of tree. These are reasonable assumptions based on the evidence available, but are ultimately mistaken because the blind men each had access to only one small part of the animal. Similarly, neuroscientists can arrive at mistaken interpretations of how the brain works if they only focus on one area in isolation. The various areas of the brain work together. Their dense interconnections support conversations between brain areas. If we only study one brain area at a time, then we only hear one side of that conversation. We all know how difficult it is to understand what a telephone conversation is about if we only hear one side of it. The same goes for brain networks. We need to hear both sides of the conversation. And with the expanded field of view that our technology provides, we now can. What you see here is a video of neural activity in the brain of a living mouse. Each one of these blobs is a neuron. When the neuron is active, this is called spiking, it lights up. We have genetically engineered the mice so that their neurons light up like when they're active like this. This imaging method is called two-photon imaging. This data is fascinating because we are observing a working brain as it processes information in real time, and we can see when each neuron is active. The resolution, being able to see each neuron, is critical. Each neuron in the brain exhibits a remarkable degree of autonomy. It makes specific connections to other neurons, and most neurons in our brains have been with us our entire lives. Complex dynamics play out over intricate networks that span the entire brain and underlie our thoughts and behaviors. Two-photon imaging gives us the resolution to see that in action. This neuron-level resolution, this level of detail, helps us develop accurate mathematical models to better understand how the brain works and what might go wrong in complex neurological diseases like autism and schizophrenia. For example, I myself cannot understand how your home congressional districts are simply by looking at a few polling numbers. You represent your districts by knowing both a wide array of your constituents and detailed information about them, including their lives and concerns. It is through that extensive and intimate knowledge that you learn what makes your communities work. Similarly, neuroscientists must study the brain in high detail and across a broad area. Now, you can buy an imaging system on the market today that will give you a video like this, but you can't zoom out. All you get is this peephole view into the brain. If you try to zoom out using a lower magnification, the neurons blur together and you can no longer tell what each individual neuron is doing. Let me give you a sense of scale for what this peephole view is. The width of this field of view is about 1/4 of a millimeter, or roughly the width of a few hairs. So this is a very small portion of the brain, even for a mouse. Before we developed our system, scientists could only look at neuronal activity through a peephole like this. We wanted to break free of this peephole view and actually obtain a panoramic view so that we could actually see multiple brain areas and listen in on both sides of the conversation. For example, my lab is currently working on understanding how the mouse brain processes vision, and many of the important brain areas for vision are here close together, but still outside of this peephole view. When I set out to address this topic, the technology did not exist to zoom out and still deliver high-resolution neuronal activity. I figured out what was limiting the current technology, and then I devised a plan to develop that technology, and in my lab, we invented it. Not everybody thought that we could do this. One grant reviewer for HFSP, the Human Frontier Science Program, wrote in their review of our grant, "It is impossible. If it were possible to design a wide-field two-photon system that retains high spatial resolution, major companies would have done it by now because demand certainly exists." Granted, this level of engineering is typically outside the expertise of many biologists, but we're a unique team. For example, I'm a neuroscientist and a member of the Society for Neuroscience, one of today's sponsors, but I'm also a member of the Optical Society, a very different field and another of today's sponsors. I and my team have degrees in physics, mathematics, optics, and engineering, as well as neuroscience, and this combination of expertise lets us rapidly develop novel and powerful instrumentation for neuroscience. Fortunately, other reviewers recognized that, and I was ultimately awarded the HFSP grant, and that work laid the groundwork for the project that is now funded by the NSF, which has an excellent track record for supporting this kind of interdisciplinary science. To build this new microscope, we had to start from scratch. We couldn't modify an existing microscope because they're so far from what we need. For example, America was already making the best automobiles in the world in the 1960s, but when NASA needed something to drive on the moon, the engineers had to go back to the drawing board and start from scratch to build the lunar rover. So to make this instrument, we went back to the drawing board as well and started from basic principles. Through extensive optical engineering, computer-aided optimization, custom fabrication, and adapted high-speed electronics, we arrived at a design and we built it. We call it the TREPPIN 2P, which is short for Twin-Region Panoramic Two-Photon. And this is just a couple of the layouts of the optics and the beam pathways. This is what it looks like in the lab. There's a front view on the left and a back view on the right. And let me tell you, this system worked better than I expected. We increased the field of view 100-fold. This is quite literally like going from a 1 megapixel camera to a 100 megapixel camera. The difference between TV in the 1980s and the latest super high-def 4K TVs is about 25-fold. We obtained a 100-fold increase. And I want to show you one of the first movies that we took with this system, but first let me remind you what the state of the art used to be. This is the field of view compared to 1/2 of the mouse's brain. This is the peephole view that we were limited to. Now see what we can do with this new system. This is some of the first data that we obtained. On the right, the field of view is indicated, and you can see how many brain areas are within the field of view. On the left is a portion of the video that is being zoomed in and out, so you can see that the system resolves individual neurons. Instead of just seeing one portion of one brain area, we can now see a lot more. Not just an entire brain area, not even just a couple of brain areas, but nearly all the cortical brain areas that are important for vision. And importantly, we can resolve individual neurons in each one of these areas. This is like Google Maps. We can zoom in and out, and the resolution that we want is there. The black regions are the blood vessels. We can't see through those, at least not yet. I wish I could share with you the sheer exhilaration we felt in the lab the day that we got this data. It was like taking the blinders off for the first time. Suddenly there was a flood of new experiments that we could now perform. We can truly examine how vision develops in the brain through conversations between different brain areas. So let me give you an idea of what we're exploring at the moment. In my lab, we are focused on the parts of the brain that process vision. Mammalian brains, from humans all the way down to the humble lab mouse, process visual input faster and more efficiently than any man-made device. If we can uncover the computational principles that are implemented in neural circuits for vision, then we can gain insights about how other parts of the brain work, and we can learn how to build better computers ourselves. When you see things, those images are analyzed by several different brain areas in parallel. These brain areas can be broadly classified into two groups. The first group of areas try to figure out what you see. Is that person your chief of staff, or is it the sandwich delivery person? The second group of areas try to figure out where objects are and how they're moving relative to the viewer. Do I need to run faster to catch that football that's headed towards me? This division of labor with different brain areas for recognizing objects and analyzing motion tell us a lot about how neural circuitry computes. And with the TREPPIN 2P, we can now zoom in and out on brain circuitry and analyze brain activity at multiple scales. We can zoom in on a local population of neurons in one area and examine how they process this information. Then we can zoom out and see how that compares with brain areas around it. We can also simultaneously image neurons in different regions and estimate what information they are exchanging. In our preliminary experiments, we have measured, for the first time, how these conversations between brain areas change depending on what the animal is looking at. There are sometimes that the neurons in two particular areas exchange a lot of information, and sometimes they exchange less. So in summary, we have created a new microscope for analyzing brain function called the TREPPIN 2P. With it, we can now zoom in and out to view brain function at different scales, increasing our ability to view brain activity by 100-fold. The TREPIN-2P enables us to better understand the complex patterns and conversations among neurons in the brain, and we are currently using the system to examine how different parts of the brain work together to support vision. Thank you very much.
Thank you so much. Dr. Oliva.
Let's move to the human brain. Let's move to talk about us. The human brain is a super high computing machine. It actually works with the same power than the light bulb in your fridge. And let's take vision as an example of what we can do. So in just a millisecond, images you see in the world are going to be printed on your retina and sent to the back of your brain, which are the visual areas. And there a lot of calculation are happening. So let me show you with a demo the power, what it feels to see and understand in only half a second. And then I will show you how the human brain understands what it sees in half a second. Those goes by fast, but you had the feeling to see every single one of them. So here is a movie about a human brain. Look at the clock. It evolves in milliseconds. And you're going to see the dynamics in the human brain, starting around 60 milliseconds after image presentation. Here are the area calculating what you are seeing. Before 100 milliseconds, before even you can think of it, you have all those activities going on. And here we are capturing activity in the human brain as you are seeing. At 150 milliseconds, you have no consciousness. You cannot move. You can even not move your eyes. Your brain is doing all the work for you, processing those images. You're still stuck in time, frozen, looking at that photo, 250 milliseconds coming. You can still not move your eyes. You might have just a slight motion, a reflective motion of the eye around 250, 300 milliseconds. So here is the brain working in time while you are frozen in half a second. And it's going to deliver to you, after a few hundred milliseconds, this feeling that you could see all at once the photo of Marilyn Monroe, the Capitol, and the other scene I've shown you. So, using two very well-known technologies that allow to record activities of the human brain in space and time, we have developed in my group new analytic tools in order to combine information at the level of the millisecond and millimeter to produce those movies. So what we can do with a human subject is you can record using MEG the activity of the brain every millisecond. But then you can also record the activity of the brain in space using functional MRI. And then you combine those with those fusion methods that we have been putting together. So now what's very exciting is that as new neurotechnology are going to be developed in the next few years, well, you can imagine that the precision at which we are going to be able to look at what the brain does over time when we see, feel, hear, remember imaging, this is going to be possible. So there are many mysteries about the human brain itself that we need to unravel, and I hope the next decade will give us a lot of knowledge about this. But I would like to share with you a couple of principles that I believe are at the core of the capacity of the human brain to evolve in an ever-changing environment, and those are also topics we are working on in my lab. So one of the first principles is plasticity. In a way, for the human brain, nothing will be lost. Everything will be transformed. This is plasticity. What does this mean? Well, let's say you have an unused terrain, a field of neurons. Well, plasticity means that a collection of neurons can be trained or retrained to do something different than what they were designed to do originally. And one of the most striking examples of plasticity in the human brain actually the brain of blind people. So this is illustrated on the slide here. If you are touching, if a blind person touches a surface, we see activity in the top part of the brain, the premotor areas. But you can also see on that slide, in the back of the brain, the visual neurons are responding. How come? Well, actually, those neurons are not visual neurons. They are located in your visual area or the visual area of the sighted people, but they have been recruited for tactile, hearing, and even reading informations. So this fabulous ability of plasticity allow the network to get resources where they are and not lose resources. A second illustration of what makes the human brain such a high computing machine, I'm going to explain it to you with example that we are faced with every day. So we are in an ever-changing environment. You have novelty every day. It's my first time in this room and on Capitol Hill. So let's look at some facts. In this ever-changing place, things move in time. So when you're looking at the world, there's some action that are going to happen in front of you at an immediate time scale, or a short time scale, or a longer time scale. You're in this restaurant ordering food. So how does the brain understand and adapt to this ever-changing environment and all the novelty that are coming to you? Well, there is a structure well protected in the middle of the brain. It's called the hippocampus. We actually have two, one on the left, one on the right. And this hippocampus is actually like a time machine of the mind. What does a time machine do? A time machine allows you to go back to the past, retrieve memories, and this is how you know that you have been there before and you recognize your friend and the places where you have been. But a time machine allows you to also see the future or imagine what comes next. And this will allow you to predict and think about what you're going to do after this briefing. So those two components of retrieving memories from the past and projecting imaging into the future are processed and held in those regions at the center of the brain, with the hippocampus being like the motor of this. And because the world is an ever-changing environment, you are facing new memories that you want to encode every day. You have a new job, new people, new friends, new places. So the hippocampus do something very important that is at the core of this high computing ability. At some point, you need more resources, more capacity, while the hippocampus grow new neurons. It's called neurogenesis. It gives the ability to encode new memories and relay them to the one you already have from your past. So what makes the brain also a super high computing machine is this capacity that nothing is fixed. The capacity can grow, and it has the machinery to grow new neurons so you can pick up new experience and adapt in an ever-changing world. So the human brain is a powerful, fabulous computing organ, and the basic research on the dynamics of the human brain is really fundamental in order to determine the sequencing and the duration for perception, memory, prediction, so that we can understand why and how this fabulous organ can be so powerful with so little energy. And I believe that all neuroscience, all of us, we could have far-reaching benefits at the level of the human brain and our well-being, at the level of adults, at the level of humanity even, for learning, healthy cognition, professional competitiveness. There's a lot of things we can do studying the human brain and neuroscience in general. Thank you very much.
Thank you, Aude.